in the last group, they are flat and they are dorsiventral. They are flattened on the surface, and so they are still very thalloid-like. They are a gametophyte. That's a gametophyte, meaning it's the haploid body. And it can be lobed. So you can get various lobing. We saw in the other, the kind of lobing was the bilateral type. Here we have lobing that can vary. Now, we have a number of things that may be a bit different. We have rhizoids are present, and the rhizoids are usually tuberculate. That's the kind. The ones that are scale-like that we saw for the last group are not present. Now, internally, the thalloid is not subdivided into groups, into zones. We don't have stem, leaves, and roots per se. So it's not subdivided. Now, each cell will contain a chloroplast, and the chloroplasts have pyrenoid. So that is very much algae-like, like what we saw in the algae. The chloroplasts tend to have the pyrenoid. Then mucilage cavities are open. Mucilage cavities open on the ventral side. So that is on the undersurface. So they will have slime pouring out of those spores. Now, what about the diversity of the sexual organs? They are embedded within the thallus. So these are embedded within the thallus. So continuing with the characteristics, so the sex organs that are embedded, we have the antheridia, which are the male, which usually can develop singly or in a group. So if they develop in a group, then you're going to have an antheridial chamber. Now the sporophyte, now we're talking about, now we've finished about the gametophyte. Now the sporophyte may be differentiated into a foot, an intermediate zone, or what we'll call the meristematic zone, and then the capsule. So it's got a foot, a zone in between the foot and the capsule. Now, due to the presence of this zone, the sporophyte seems to grow. It can grow quite tall. So it continues to grow. So it has what we call indeterminate growth. It continues to grow. Okay, now the part that forms the spores, the archaeosporium. Now this I'll have to explain later, that origin. So it has an archaeosporium that produces the spores. Then the actual material that produces the spores, so we call it the sporogenous tissues. Now, these are the ones that form the spores, the fertile spores. And it will also produce elaters. Remember what elaters were for? Elaters are those that push out the spores when they are ready. So we have the sporogenous material or tissue that forms those spores, the fertile spores, and then the sterile elaters. Now, the capsule wall has various layers. It's 4 to 6 layers thick, and that's a new one. The epidermis has stomata. The epidermis has stomata. So it can undergo, gaseous exchange. So the capsule matures from the apex to base and usually dehisces into half. So the apex matures first before the base. So it's the top that will open up. So you get two valves. So when it's ready to dehisce, it begins from the top and breaks into two valves. Okay. I think there's somebody waiting in our waiting room. Let me just check. Okay, so continue. So this is a diagrammatic representation. So we have the gametophyte, which is the haploid part. And so these are some of the characteristics of the gametophyte. It's the simplest in the group. Actually, the gametophyte here tends to be very simple because you remember in Mercantiophyta, we had the male and female on some branches, okay? On certain structures. They would not necessarily grow within the gametophyte body itself, but they would have those umbrella-like structures on which the male and the female grow. So here we find that it's very simple because we really don't have those structures that we're talking about. Then the gametophyte, it's usually very small, thalloid, and it may have very little internal differentiation. So the internal differentiation is not much. It's not much. Then the male and the female are usually embedded in that gametophyte. The male is usually embedded on the upper portion, of the thallus, while the female, you can see is lower down. If you look at this picture, you can see this is in the upper portion, that's the antheridia, while the female tends to be a bit deeper, it's well embedded, and in direct contact with the vegetative cells. So this, you can see, it's really not in contact with vegetative cells. So this is deeper. The positioning is deeper within the thallus of the gametophyte. So this is actually also showing the life cycle. So you can see the gametophyte, and then you see the sporophyte. So the gametophyte is the one that still houses or holds or supports the sporophyte. So the sporophyte is not independent. It must survive on the gametophyte. So this is the gametophyte. This is the sporophyte. And you can see the sporophyte, we talked about the foot, then the meristematic zone, and then the capsule. So if we look again at the sporophyte, even though we've described it a bit, you can see the sporophyte tends to grow long, so it's long, rod-like. It is upright. We've already mentioned it has a foot. It has a a long cylindrical sporangium, which is embedded in the thallus. Then you can see it's a, it's a multicellular structure. So it has a multicellular outer layer. You can see it on the diagram. But the outer layer is covered with a cuticle and also stomata. Then it has a central columella. You also can see it from this particular diagram. And then it has various layers, a layer of tissue where spores are produced. So we have spores and elaters. So, okay, so that brings us to the end of the Anthoceratophyta group. Okay, so we'll move on to the next group, which are the bryophyta, the moss group, the moss. So this is the category that has the largest number of species. So species and genera, 700 genera, 14,000 species. So it's the largest class. Now, general characteristics, it has a main body that is a gametophyte, which is not new. That's what we've already seen in the other groups. The main body is the gametophyte. However, here it is differentiated into two. You have the juvenile stage, and then we have the leafy stage. How are those different? The juvenile stage is just a filamentous branch, which we call the protonema. So it's very much like the moss, the Macanthiophyta group. It is just flat, or in fact more like an algae than a bryophyte. So the juvenile stage is very primitive in its form. So it has taken up the form of the algae, where we could see filamentous branched groupings. So now this group, the juvenile stage develops after germination of the spore. So once a spore has germinated, it gives rise to the juvenile protonema. Then the protonema will then develop into the leafy gametophyte. So the leafy gametophyte is either branched or unbranched. And here it differentiates into the three parts that we know of a typical plant. So we have the rhizoids, the stem, and leaves. Now, branches arise below the leaves. You get branches and then leaves. So this is actually very plant-like, as we know plants to be. So the leaves have midribs. They are unlobed. They don't have lobing. You know, the edge of the leaf is not lobed and is usually arranged. Each leaf is arranged in a spiral manner. So they have about 3 to 8 rows on the axis. So you get a spiral leaf arrangement. Now, we still don't have roots, we still have just rhizoids, but the rhizoids now are multicellular. They are multicellular, so they look very filamentous and they can also be branched. So you can see some sort of evolutionary improvement in that area. So the rhizoids are now multicellular and branched. What about the sex organs? We're still looking at the gametophyte. So the sex organs are usually at the apex of the stem or of a branch. So they are apically arranged. Now we come to the sporophyte. The sporophyte is green during the early stages, and it can be differentiated into foot setter and capsule. So remember in the previous one that we've just looked at, the hornworts, we didn't really have a setter. We just had meristematic tissues. But now here we have a setter which is well differentiated, in addition to the foot and the capsule. So the setter is usually elongated and rigid. We have a columella. I hope I'll show a diagram there with the columella. The columella is usually present, and internal, its origin, it's made from within itself. So endothecial, from within. Now, Axporium, which is a spore-forming tissue, is differentiated only in spores. Elaters here are absent. So we've seen elaters in Mercantophyta, we've seen elaters in the hornwort. Here in the next group, they are missing. So dehiscence normally occurs by the lid. There's an operculum on the capsule. That lid normally opens up and it pushes out it causes, of course, the spores to come out. But what helps the spores to come out, there is a structure that we are going to see. It's called the peristome. That's the one that pushes out the spores from the capsule. So once the spores land on suitable ground, they will germinate to produce that protonema that we said is the juvenile stage of the gametophyte. So now looking at the gametophyte properly, so we've already talked about the two, the protonema and the mature gametophyte. So the protonema we've seen is the filamentous early stages, filamentous structure. Then we get the mature gametophyte. Now this has differentiation. So you can see it has rhizoids. Now the rhizoids are for anchorage. We also have rhizoids for water collection. So they are able to conduct water because they have certain new structures that we are calling the hydroids. So they are hydroids, they can be able to pick up water and transport it through the structure, the whole body. And then there are many chloroplasts. So the gametophyte can actually undergo photosynthesis. Now, if you look at the moss, this is the moss, it's radially symmetrical. So it's not like we saw dorsiventral structures. No, this is radially symmetrical. So it's round. So you can see all the parts are arranged in a circle from the top to the bottom. Then the rhizoids tend to be multicellular. These are the new structures that we are seeing in the moss. So it's radially symmetrical. Rhizoids are multicellular. Then it has leaves with or without a midrib. So they may or may not have midrib. So here you can see that evolutionary development. It's very plant-like. Now, the sporophyte of the moss, of the bryophytes, is more advanced compared to what we saw. So the sporophyte has the following. So we will have the capsule, and the capsule is basically the sporangium. The sporangium is a spore-producing body. But we don't call it a sporangium because it has other structures on it that make it different from the normal sporangium like you see in fungi. This is more developed, and it has other structures that help to keep it safe and may ensure that it does not undergo dehydration. Then it has a seta. So we've seen the capsule, which is, if they look in the diagram, that is the capsule. Then we have the seta, which is this portion just below the capsule. And then right at the very base, it's not seen on this particular picture, is a foot. And the foot penetrates the thallus, the gametophyte. So it penetrates into the gametophyte. Then on this capsule, we have various structures. So we have the calyptra, which is the top of the archegonium, and its work is to protect, actually at the top of this structure, which is it's not seen here. It's been removed. So the calyptra is the one that protects the capsule. And then below the calyptra, we'll have the operculum. So that is what you see here. We have the operculum. Something here that is blocking me. Okay, yes. So we have the operculum. That operculum is the lid of the capsule, and it's actually found underneath the calyptra. On the sides of the operculum, we have the annulus. The annulus are a group of cells that are found at the base of the operculum, and their function is to open that operculum. So those annular cells are able to break up, and once they break up, they release the operculum. So in that way, opening up the capsule. So they are associated with the opening up of the capsule. Now we have, just below, we have a peristoma. The peristoma are teeth-like projections. What they do is that they are able to straighten out when dry. So when they are dry, they straighten out and they lift pollen out of the capsule. So usually found at the almost at the top there. Then we have the columella. This is a sterile portion. This portion here is sterile portion of the capsule, and its function is basically to support that whole capsule, giving it food and ensuring that the spores are undergoing development. So these are more examples of the way the capsule look. So you can see the trying to look for the calyptra, which I believe is this. Then we have the operculum, should be with inside here. You can see the columella. This is how it looks in real, not just diagrammatic, but the way it really looks in the plant. And then the peristome teeth are actually shown here. These are how they look. So they are found underneath the operculum. And so when they straighten out, then they push out the spores. And then below you can see the septa. So actually in this diagram here, that's where you can actually see the calyptra very clearly. So you see the calyptra, which is a cap. It's like a hat on top of the capsule. And that can break off when, when needed, when it's mature. It can break off. Okay, now let's look at the reproductive structures of the moss. So here we still have antheridia and archegonia. So let's look at the male first, the antheridia. So antheridia are pear-shaped. They are pear-shaped, and their function is to give rise to the sperms or their anthrozoids. But here we have they are born on a head. So they are born on an antheridial head. So this is the antheridial head, and inside this head are very, very many antheridia. So each of these are the antheridia. And the antheridia are interspaced by paraphyses. These are just hair-like structures that grow in between. Their function is really to support and to cushion the antheridia. You can see they are pear-shaped structures. Now, archegonia, female, the structures themselves, the archegonia, are flask-shaped. We already saw that in the previous group. They are flask-shaped, and they have an enlarged basal portion that we call the venter. So again, this would be it. That portion is the venter. Then there's an elongated neck before it comes to the atmosphere. The neck usually has cells that completely, close up that entrance until the right time. So when the right time comes, then those cells in the neck will break up, and that space will be filled with water, and that will enable the sperm cells to swim into the neck, down the neck to the venter, and cause fertilization to take place. Okay, the general life cycle of the moss. So if we begin, let's begin at this point where the spores have formed from the sporophyte, from the capsule. So you get the spores, and the spores germinate. And because we have male and female structures, we have male and female plants. So you'll get a male gametophyte, you get a female gametophyte. So some spores will give rise to the male, others will give rise to the female. And you get, first of all, the protonema. So when there are many protonemata, so male and female So the female will give rise to female organs, and the male gametophyte will give rise to the male organ. So we get sperm, we get egg, fertilization happens within the venter, and you get the zygote. The zygote will develop into a sporophyte, and the sporophyte matures. So you'll get the capsule. This is the capsule, and we've already looked at that structure. We have the capsule, we have the operculum, we have the columella within, and all that. Eventually, it matures to give rise to the spores, and the whole cycle begins again. So that is the life cycle would repeat. Let's look at the economic importance of the bryophyte. So this is general and looking from Mercantophyta all the way to the moss. What are the economic uses? So one, they're important as fuel. Why? Because of the fact that they decompose very slowly, they're able to provide a fuel that we call peat, which is quite valuable. This is especially so in those very cold areas, because then the decomposition is going to be very slow because of the cold weather. Now, secondly, there are good indicators, bioindicators of pollution and environmental degradation. So once their environment changes, then they will undergo, degrade. They will die, or they will not grow well. Now, one of the reasons is that they lack the cuticle. If you remember the cuticle, we found that only in the sporophyte, which is a shorter living body. But the gametophyte does not have a cuticle, so it can lose water easily. So it's a bioindicator of pollution and environmental degradation. So we also have bryophytes that are useful as medicines. So for example, minium is used to treat burns and bruises. Then they also make very good bandage material. Why? Because they can absorb water or fluids to 35 times their dry weight. So you can use them as, the napkins or bandages. Then they are good as packaging because they are soft, they are spongy, so they can be used to package material. Then bryophytes are important. We use them for soil erosion prevention, because why? They form a thick layer, mat, over soil. So when it rains and wind comes, it will not carry away the soil. Then another economic importance is that they also form symbiotic relationships with other organisms. For example, they can combine with cyanobacteria and be able to increase nitrogen in the soil. Then they are good experimental plants, because first, they're easy to propagate. You can grow them very easily. They can grow in very small spaces. You can clone them and get genetic replicants. And then, because they are haploid, you can, they're haploid, so they only have one set of chromosomes. So you can actually see what happens if one chromosome is affected, if the mutation happens, you will see immediately what the phenotype or the reaction will be of that plant, which is difficult for those plants that are diploid because the other chromosome, its counterpart, can mask the mutation, so you don't see the effect. But because they are haploid, you will be able to see whatever genetic alterations have happened in the phenotype. Okay, this is something for you to look at, some of the differences between liverworts and moss. You can also do a table like this and just show also the hornworts and liverworts, what are their differences, and then also between the hornworts and moss. You can check the differences. And that brings us to the end of that particular lecture. Any question? Okay, I think we will stop there and we will continue with the bryophytes in the next lecture. I'm trying to organize that we have a practical on Friday. So I would request that you be here on Friday so that because practicals are physical, so you need to be here. So we normally meet in the general lab. The general lab is in the old building, in the old, block 2. Towards, as you enter into the block 2 from, climbing up, it's on your left, on the zoology side. So you go towards the zoology side, the general lab. So we'll set up for you. So please ensure that you are there for the practical.
What time?
Yes?
Time.
9 o'clock. We start at 9. So you'll be there by 9. 9 o'clock. Is that clear?
Yes.
Fine.
Yeah.
So yes, you'll be given your material, you'll be given your handout. If possible, is it Sarah? Sarah, you're the rep. Please come and liaise so that we get the handout so that you can circulate to members. Okay.
What
So
about
come on
notes?
Thursday so that you get the handout from Mutiso. Do you know Mutiso? The technician. Yes. So you'll come and get the handout from Mutiso so that you can circulate to members.
Yes.
Notes.
So we'll meet on Friday, 9 o'clock in the general lab. 9 o'clock.